Okay, thank you very much for this lovely invitation to speak to you guys today. I wanted to share with you some of our experience with extracellular vesicles. Ruching has just hot off the presses generated some very beautiful TEM images for us. And in the course of discussing generating those TEM images, we were talking about how difficult it can be to characterize extracellular vesicles. And so I wanted to share with you some of the lessons that we've learned that were, painful and hard won, but have become things that we're sharing with the field, and the field is beginning to adapt in the context of extravesical studies. Everything I'm going to show you today, really I have to give all of the credit to Jubin George, Michelle Pleat, and Joshua Welsh, who were really the driving forces behind components of this that I'm going to share with you today. And so one of the things to really understand about extracellular vesicles and why they are so very, very complicated to study is, as you all know, they're very small, but they're also very heterogeneous. And so there are large ones, there are small ones. We now recognize extracellular particles that are important parts of this kind of extracellular milieu that are different from viruses, that are different from lipoproteins, and from other things that may be found in biofluids. So the nomenclature in the field has been evolving as we've learned more about what it is that we're seeing and understanding better the diversity of the different types of particles. This is a very, very high-level, simplified overview, building on kind of how we saw extracellular vesicles as exosomes and microvesicles 10, 15 years ago. But this has changed a lot. And so it moved to referring to these lipid bilayer bound extracellular particles as extracellular vesicles, to be somewhat agnostic as to the biogenesis origin of the vesicles. And as the additional diversity and identification of exomeres by David Leiden's group, particles that are not lipid bilayer bound, but they do contain lipids and proteins and nucleic acids, and tend to be smaller than extracellular vesicles. And then supermeres, which are also multi-component condensates of lipids and proteins and nucleic acids that are extracellular. So these are all extracellular things. And now the most recent MISEF sort of suggests that we call this family of extracellular entities extracellular particles, because when we're looking at EMs, when we're looking at nanoparticle tracking analysis, whatever it is that we do to look at these things, we're going to refer to them as particles, and then we can subdivide them based on whether or not they're vesicular. Do they have a lipid bilayer around them, or are they non-vesicular extracellular particles? So I think this is, people will often say, well, what does the nomenclature matter? And I think it matters in a couple of ways. One, so that we have a harmonized way of understanding each other's results more clearly, but also so that we can dive into the different biologies that these, the different biological pathways and networks that these are playing into, because they do have very beautiful and striking differences if you look back at the papers from Bob Coffey's group, David Leiden's group, and others. So I'm a radiation oncologist, and I'm interested in the interplay between cancers, radiation treatments, and the immune system. And so to me, one of the great strengths of the extracellular vesicle field in terms of its potential is that we could potentially look at a patient biofluid sample, and if we can identify immune EVs, immune cell-derived EVs, tumor cell-derived EVs, and vascular cell-derived EVs, you could envision an approach of looking at subsets and how those cells in the patient are responding in real time, since EVs turn over at a rate of a couple times a day, most of them, this this is a promising approach. However, that's not the approach that's being taken really widely by the field. In general, the liquid biopsy methods that look at these extracellular particles tend to take a biofluid, isolate the exosomes or extracellular vesicles in bulk, and then isolate materials from that, and then identify biomarkers. So what I'm going to share with you is our journey to try to be able to meticulously parse the subset so that we can study the cargo and biological functions of the different extracellular particles. So the original intent that I had was to do RNA-seq of the separate subsets. And, in order to be able to parse the subsets, I knew I needed to reliably identify the different subsets and then be able to assay those different subsets with a highly sensitive analysis approach. So this talk is really going to focus on how do we identify those subsets. And I started this work when I was at Stanford before I came to NIH with Marty Bigos and colleagues there, using an influx flow cytometer where we were able to look at small particles, not as small as we might have liked, but small particles because of a small particle adapter on the flow cytometer there. In the process of doing that, though, we realized that there was a great deal of day-to-day variation, a great deal of fiddliness with setting up the instrument and reproducibility. But in doing that and coming here and working with additional instruments since then, we developed a method that we called NanoFax, where we could consistently set up the instruments in a way which we could see some particles above the background noise, which we called reference noise. And this is really different from how people will run flow cytometers. Typically you set thresholds and background gates so you ignore the background noise. But we would set the thresholds at a level at which we could see just enough noise to balance with the flow rate of our samples so that we could know where the noise was and where the samples were running relative to it. That study we published in JEV in 2019 and also 2017 in JCI Insight. And with that approach, we sorted viral vesicles, viral subsets, essentially. And so what we did was we took a mixture of different HIV types and then labeled them either red or green, and then sorted them, and then took the sorted material, reanalyzed it to assess purity. And so you're seeing here the sort of the mixed sample, and then the reanalysis of the isolated material. So what you're seeing, these are the red ones, and this is the reference noise that I mentioned. And these are the green ones, and this is the reference noise. And what we showed is that you can take these and put these on cells that have specific tropism for these particular viral types. And you can see that the reporter line confirms the fidelity of these sorts. Those which are, CCR5 tropic, in fact, CCR5 cells, and those that are CXCR4 tropic infect only the CXCR4 positive cells. So this was an elegant solution, and I have a lot of people who will ask me, well, what about using this sorting method preparatively in GMP production of EVs and that kind of thing? The caution here is you're sorting one particle per droplet, and the particle volume, since these are roughly 100 nanometers, relative to the droplet size, is a gargantuan difference, and you really sort liters to get appreciable quantities, or hundreds of milliliters to get appreciable quantities of material. So it's not a practical solution for, I would say, EV production, but it proves that we can begin to use flow cytometry to approach these things. And so as we were wanting to collate this information about EV subsets, we realized we needed an infrastructure that allowed us to catalog the surface proteome, intraluminal cargo features of the EVs that we learned based on microscopy and flow cytometry and other attributes like concentration. So this is really the backbone of Josh Welch's work when he was here in the lab as a postdoctoral fellow and staff scientist. And he took the challenges of the field, the arbitrary units of flow cytometry, the lack of calibration tools, a lack of a reporting framework, and he developed solutions for them, which we're really quite proud of, that have provided an infrastructure for the field to build on. And then we also expanded into bead-based assays to have a more dexterous way of evaluating the diversity of EVs that are in our samples. So again, back to this is the overarching goal, and this relates to our patients. So we need this to be reproducible. So we kind of prioritized at the top of the priority list to move forward with this research program. We need to be sure that we're making precise and reproducible measurements. These are multi-component particles or vesicles, whichever it is that we're studying. And so we need multi-parametric assays. We need standard units so we can reproduce results between labs. Then we have to be able to integrate the data in a cohesive way. He first tackled single particle scattering. This was his passion from his PhD. He developed a number of tools, but then paired with Edwin van der Pol and a consortium that we formed to introduce a lot of the specifics of this in extensive detail. So if your colleagues, collaborators, or group are interested in studying extracellular vesicles and are remotely considering incorporating flow cytometry into the assays, we tried to make this paper approachable so that you could learn to understand the assays that you're looking at in flow cytometry with relationship to EVs and why we do what we do. And this is the crux of the difficult thing to understand, which is just simply that most of our instruments that are optical, EM aside, just with light scattering or fluorescence, many of our instruments have difficulty in seeing sub 100 nanometer particles. But when you look at the population of particles that exist in our biofluids that we're interested in, there's actually many, many more that are small than that are large. So this having a low limit of detection, a small nanometer limit of detection, is really important for being able to adequately survey the breadth of things that we're looking at. And also then that applies to staining of markers, because it can be difficult to see. Most flow cytometers, the flow channels don't detect fluorophores, a low number of fluorophores. So they certainly aren't rated typically to find 1 or 2 fluorophores. But if you think about the density of how many receptors could you pack on the surface of an extracellular vesicle, this is an estimation of how quickly that falls off. When we get down to 100 nanometers, you're approaching 10 particles or fewer. So you need instruments that can detect 10 particles or fewer. And further complicating this with flow cytometry, from the beginning flow cytometry, they recognized that with me scattering, the waveforms and intensities of light detected from the flow cytometer that are refracted off of the scattered off of the particle are nonlinear. So modeling this is quite complicated. And I have to say we have relied on, and I apologize, this came through a little funny looking, we've relied very heavily on our TEM images as our gold standard of size and distribution and basic surface morphology to compare to what we're seeing with our flow cytometry. Calibrating based on known calibrators is something that was developed before we really started our EV research in this area. So it was really established that it's very helpful to use MESF standards. This isn't a crazy thing that we came up just for EVs. It's a phenomenon that was recognized in the flow field to improve consistency between data measurements. These are the events uncalibrated, and these are calibrated in MESF units, which allows you to derive conclusions about what you're seeing. And so we then applied this to our EV work. In 2015, we formed a joint working group to really try to come together to not reinvent the wheel. ISTH, because of their study of microparticles from platelets, had a dedicated interest in EV research. ISAC, as a flow cytometry group, did, and ISEF did. So we came together and we said, let's all work together on a working group for this. John Nolan then also kind of pointed out, well, maybe we can get around the scattering challenges by fluorescently labeling our EVs and using the fluorescent labeling as a surrogate for size. And the challenge with that is not all of our particles label in the same way with the fluorophores. Let me jump ahead a little bit to a work that was really kind of led by Vera and Josh to apply these calibration standards and show how if you do standardized measurements and assessment and reporting, it enables interlab reporting. So Vera was at the University of Ottawa, and Josh here. And what they did was together they looked at cytometer A and cytometer B, and this is the typical data that we would get with uncalibrated detectors and different instruments in different labs, or even the same instrument in the same lab run with two slightly different settings. This is what you might see. These are arbitrary units. You can certainly see there are distinctive populations and patterns, but it's difficult to relate one to the other. By calibrating them, we bring them into a harmonized frame of reference so that you can say, okay, this limit of detection here is a bit higher, so we're losing some of the smaller particles with this instrument that are able to be resolved here. And that actually helps draw that conclusion and be more specific about what we're seeing. You know, this really helps when we're presenting data. And some of the data I'm going to show you later, I need you to remember this is why we focus on calibrating across our assays. Then we published this working group paper as a guideline for reporting. How with the challenges of these types of data analysis, how should we be reporting our data? And we developed a framework that provides this framework for elements of the work to be reported. So not only the primary data that we later developed a flow repository where people can put that primary data to be analyzed by others, but also these details of the experiment to go along with the data so people could compare to their own results. I'm going to skip ahead to an example where we were looking at EVs, and I just want to show you why TEM is such a valuable tool for us. We could see there was a distinctive population here, but in looking at TEM, we can see these are vesicles. They have this characteristic dimple. There are some particles here which are extremely small, which, you know, we could ask the question now that we know about exomeres and suprameres. Are these exomeres and suprameres in our preparation? That's quite possible. We took the material and we sent it for cryo-EM, and this is what came back. And this led to another era of our lab's research. But what you can see is that unexpectedly, we actually see these icosahedral shapes consistent with viral capsids. And this is a mouse cell line, an immortalized mouse cell line, which is sold by a well-known commercial vendor as a BSL-1 cell line, and had no known pathogens associated with it. We have since sequenced this and have characterized it as a known ecotropic mouse virus. So this isn't going to infect humans in any way, but this is there, and it's important to know that it's there when working with other mouse cell studies for sure. So this is beautiful, and you can see the lipid bilayers. You can see some of the sort of naked capsids there. You can see we also have some of this stuff in the background. And, you know, you guys are so much more experienced at EM. I'd be happy for feedback of what that might appear to be to you. We haven't been able to ascertain exactly what that is. One of the leading theories of our collaborators is that this likely represents lice, extracellular vesicle, cargo, and internal contents, which happens during rupture of vesicles during the cryo process. So that seems plausible, but if this has features that you recognize as potentially something else, I'd love to know. And then you also do see these vesicles within vesicles. So these were taken from a sample that was generated very early on in my lab when we were still using ultracentrifugation as a main method of serial ultracentrifugation and isolation of vesicles. So we subsequently published this compendium, which is the great resource that I recommended that has everything from theory to specifics. And it's really a small textbook. It's about 80 pages long, and recommended reading for anybody wanting to jump into single vesicle flow cytometry. Beyond single vesicle studies, we decided that, you know, this is very complicated, and given the constraints of single vesicle studies, we're not really cracking the diversity problem of the extracellular vesicles that we want to look at. So we pivoted to a bead-based approach, and we're using a commercial bead-based assay, which is widely used and commercially available, and you'll find it in our publications. But we use that as a prototype to learn to do these assays. And it's important to realize these are semi-quantitative. It has a panel, a Luminex type panel of beads, which are coated with 37 different antibodies, which then the subsets capture those 37 different types of receptors on vesicles. We wash away the unbound material, and then we stain, we detect the vesicles on the beads with a mixture of tetraspanins, which are surface receptors commonly found on vesicles, but not unique to vesicles and not, you know, diagnostic for being an extracellular vesicle. But CD63, CD81, and CD9 are common among extracellular vesicles. And so we use a detection trio BICS to detect those three tetraspanins. When we run the flow cytometry, this is what it looks like. This is what the beads look like on forward scatter and side scatter. And the nice thing is these beads are in the micron scale. So we can use a conventional flow cytometer. We do use optimized settings for sensitive detection of the detection antibodies on the beads. And I'll show you that in just a minute. We gate out the doublets, we focus on the singlets, and then you get this really nice multiplex distribution of the different beads. Here are the different assignments of the bead specificities. And because Josh is outstanding at developing these tools, he derived a way to take all of this data, which is very complicated to analyze, stitch it together, and analyze it in MPA-PAS, a software module that he created and that we have a download link for. So with this, it can generate heat maps and principal components plots so that you can see kind of the distributions of things. But logistically, I think it's important to note that to generate this data, we do first take the FCS files from the flow cytometer, and then we calibrate the data with another software package that he developed, FCMPass, and you can find the publications and references to that elsewhere. And then once it's calibrated in FCMPass using MESF or fluorescent standardization beads, then we can reanalyze it, and that then generates standard units. And with the standard units, it allows us to generate these plots. And it also has, for the same assays with the same lots of beads, and it enabled us to stitch assays across sets of samples and days and those kinds of things, which is very helpful when you're running large batches of samples. This is an example of a study that we did with extracellular vesicles from plasma, serum, and CSF. And you can see the CSF stands out on its own. It is just a beautiful biofluid to work with EVs because it lacks the lipoproteins that interfere, I think, with the EV capture and detection from plasma and serum. So we get these really nice staining patterns. And we did this study with Michelle Pleat and Steve Jacobson, who she was working with at the time, to look at CSF from patients who had HAM-TSP, an HTLV-associated myelopathy, or healthy donors. And we basically performed that same multiplex approach. One thing that we also looked at was, do we gain anything by doing neat isolation, QEV, another isolation method we call EV clean, or that was just a control there. And we found that the CSF samples did not benefit from extra protein removal. In fact, the signals were strongest and most robust if we just left the CSF alone. We cell-free CSF, but it was, the signals are quite low with CSF, so that was beneficial just to minimize processing. And with this, they detected some informative differences about the types of EVs that are there in that viral-mediated neurological disease, and they're following up those additionally. So I wanted to kind of conclude here, and I'm going to switch to an example of data from Ruching and Jubin after I just walk through these basics. Ruching had asked that I come and be available to speak to some of the best practices for isolation, because our EV preps do quite well in the TEM studies with very little background. And so I wanted just to mention what I would consider the main points about EV preparation requirements. If you're going to do something like the multiplex flow cytometry, in that case, you have a dual integral membrane detection system. The system is with one antibody detecting an antibody that is an integral membrane protein. So it's a transmembrane protein that you're detecting, And then the other antibody is detecting a different and independent antibody that is also in a lipid bilayer. So by definition, when you have detection of a positive bead in that assay, you have two transmembrane proteins held together by by a lipid bilayer. And that's not proof that we have extracellular vesicles, but it's a pretty good indication. And when we've taken those samples then back to single vesicle assays, what we have observed in terms of the patterns with the multiplex approach holds up in the single vesicle approach to the extent that we've tested it. So I think that the multiplex approach, which uses standard, like, 8 micron, 10 micron size beads that you can run on a standard flow cytometer, takes a little optimizing, but you can still run those. This offers really good direct biofluid to bead or biofluid to assay possibilities. I think it's good for screening. We're limited to the library of antibodies represented in the bead set, but it is very, very messy to do a bulk prep without isolating EVs for other kinds of assays. And I don't mean the multiplex is very messy. I mean direct to biofluid assays. There are very few that are amenable to that. For single EV assays, I really consider EM and cryo-EM still the gold standards for seeing EVs. There are some beautiful new technologies with STORM, SQMLM microscopy, looking at single molecule blinking with fluorescence to look at EVs. But even with that, you're looking across a population of diversity, and just to see the structure of the vesicles, EM is, you see what you got. It's really wonderful. But I think to get good EMs of EVs, you do need to do isolation. So different labs have their different processes. Juven George, who's been in my group for 3 years now, has become the master of large-scale production of extracellular vesicles. He really is our in-lab mini GMP, pseudo GMP production facility for bulk EVs that we use for many assays. And the approach is we're working with tumor cell lines, so this kind of scale is possible. We expand them until they're ready to go into 10 to 20 T225 flasks. To produce the EVs, we need to produce that in serum-free media. And the EV production, we leave them in serum-free media for 48 to 72 hours. So we tend to screen each of the cell lines we want to do the productions with against different serum-free media options to identify those that disturb the cells the least. And Optimum is a good one that we use for most of our adherent and tumor cells. For some of our suspension cells, we've been using ex vivo, which some cells find that seems to be more amenable to their growth. Then we'll harvest all of these, do the requisite checking of cell viability so that we can make calculations of vesicles produced per cell, and remove the dead cell, remove the cells, remove any fragments, and also, unfortunately, we have had to choose to focus. So we're focusing on small extracellular vesicles, and we do 2 additional either 2,500 G spins or 5,000 G spins to remove the very large vesicles. Then once we have that supernatant, and this is on a liter scale of supernatant, so 500 mils to 1,000 mils of supernatant, then we concentrate the solution down to 5 to 10 milliliters with omega ultrafiltration membranes. So we spin those to do the concentration, and then once concentrated, then we take the concentrate, which is protein depleted, but not yet pure EVs without all the extra proteins. And then we clean it up on size exclusion chromatography columns. So the size exclusion approach was developed in Teresa Whiteside's lab and also independently in Rank Newland's lab about 10 years ago. And there are commercial options that make that reproducible as well. So yeah, this nomenclature dilemma is pretty contentious in the field, but I think the thing is to some investigators and groups, exosomes do have that meaning of going through an escort process through intraluminal vesicles and being released in that way, whereas microvesicles might be shed from the surface. That was how there was that understanding, but then there was the realization that there are so many different types and so many different routes of release that when we're looking at a cell culture supernatant or a biofluid or just about any sample with extracellular vesicles, unless we are you guys looking at an EM of the cell in the process of doing the thing, it's really hard to say what it is. All we can really say is it's a particle. And unfortunately with TEM, although I love TEM and it's the most beautiful way for us to look at our samples and see what we see, you're right that there are small particles as well in our isolates, and it's difficult to say if, are some of these small particles very small vesicles, or could these be exomeres? And the definition of that boils down to, do they have a lipid bilayer? And that's not something I can ascertain from this. So then we need cryo-EM. And I can tell you that cryo-EM is so difficult for most investigators to get that when we wanted to go and do follow-up studies for cryo-EM studies, cryo-EM at a really excellent outside commercial vendor that we worked with to get some beautiful cryo-EM images. I didn't show those today, but that's getting those images is thousands of dollars per sample, which is way beyond the budget of most of us in academic research. So, I think building the wisdom and the infrastructure and experience that Ruching and others of you in the group have for working with EM for EVs, and expanding that to really include robust resources for cryo-EM would be a huge benefit to the field, because we do need the both end approach of looking at cryo-EM and TEM. And just to get these in this kind of way that we have them, I think, Rucheng, you tested 8 or maybe 10 different negative staining methods to really optimize how do we best, sort of see the contours of this. But when you do have a well-purified sample, then it becomes really a beautiful image. We're going to be very proud to put these in a publication that we're working on soon. So anyway, I'll stop sharing there and take any other questions that you have.
Well, Jennifer, thank you. This is very informative. As you know, some of the colleagues already mentioned that we all have worked with EV or exosome. We don't really know the differences because sample comes and goes. We don't always get to follow the research. So this is really great to hear about, the research part that and how you are using it and how it can help with biomedical research. Any questions for Jennifer? Please go ahead.
Sorry, I still have one more question for Jennifer. And basically, as you show for the workflow, I think that the how to isolate the extracellular vesicle, all the exosome, is still different from the sample sent to me so far. I think a majority of the people use commercial kit, which I really don't like it. Whenever they use a commercial kits, and it's likely I don't see the vesicles, right? They can do whatever protein they can find, but the vesicle is hard to see. So to me, and the majority of the people, I still like the way they do the ultracentrifuge, like a traditional way. So to be sure, we can see the extracellular vesicles. But with the output from your pictures, you got a really concentrated, nice exosome. So that's become your standard method to use those columns?
It does. So, you know, those commercial avenues for isolating EVs, I can tell you there are two common ones that come to mind. One is a something like ExoQuick, or sort of a precipitation-based method that is based on an old protocol that was, you know, it is still widely used for precipitating viruses. And then you get kind of an aggregate of clumpy stuff aggregated together, and it doesn't really perfectly wash out the proteins. You get a lot of stickiness to things, and we actually had trouble getting those aggregates to come apart. We found that we had some trouble getting antibodies to bind well. So I don't think it's an entirely inert process, and that's why I stayed away from that at the beginning. Even though it's so helpful to concentrate, if you're looking at characterizing and phenotyping them, it can be challenging. There are also column chromatography affinity type methods where you kind of load it on and wash, almost like a you know, DNA isolation column type of thing. I really can't speak to those. I had such a bad experience with the other kind of kit-based approaches. I really wanted to know how what was happening to the vesicles through the process so I could make sure they were staying intact. So the compromise that we've made is that we realize if we're going to do things like Dolores Divisio and study large oncosomes and the big vesicles that get blubbed off, you know, in that case, we need to do low speed spins, and we need a whole different protocol. So if you're interested in those, or you work with folks that's interested in those, talk to Dolores Divisio and others. If you're working with small extracellular vesicles like we are, using a concentration method first, an ultrafiltration method first, is it kills two birds with one stone. You concentrate and you remove some proteins. So then the dilemma is do you go with a 100 kilodalton cutoff or a 300 kilodalton cutoff? And for our small EVs, we use 100 kilodalton, but that means, man, our poor trainees are there like hours and hours during the centrifugations to babysit those concentrator spins. And we will concentrate genuinely like 100-fold, at least 100-fold concentration. And then in that process of ultrafiltration, which concentrates, you also have not a perfect depletion of, removal of proteins, because there are still some proteins that are bigger than 100 kilodaltons. And so, you concentrate down, you remove some proteins, and then you run that concentrate over a size exclusion column. For these tissue cultures, we can reuse these size exclusion columns through washing protocols. But what we've and others have found is that if you're doing serum samples or plasma samples, you could it becomes impractical to reuse size exclusion columns, because you can wash till the cows come home. It seems like you just wash hours and hours, and you still have residual lipoproteins left on the column. So some vendors do sell single-use little size exclusion columns to use. So I'd recommend looking into that if you have folks who really want to dive into purified EVs.
Thank you.
As a last thing, for your investigators who do use ultracentrifugation, two little words of wisdom about that. We're learning that the ultracentrifugation process does distort the EVs a little bit. It can kind of bend them. It can cause EVs inside EVs. That's okay sometimes for what you're looking at. If that's okay, and you want to go that way, and you still want to avoid the criticism of, oh, but there's a lot of protein that comes down with your EVs. Robert Rafai has published a really beautiful protocol where they do the serial ultracentrifugation, and then they take the pellet and then they float the vesicles off of the pellet in a little kind of gradient system. So you've got the layer where you can recover the vesicles in a concentrated way, but the proteins stay pelleted. So I would look up Robert Rafai's paper on that from about 2 or 3 years ago. It's a very nice method.